Hello everyone, I am Rizka Alifa. This is a learning video about virus. Assalamualaikum warahmatullahi wabarakatuh. Good morning class. How are you today? Okay, that's good. I'm so happy here you all and the good condition. You are so amazing. Before we start, we have to pray to our belief. Please, Chairman. Okay, thank you. Go ahead. Okay, so this is my first class to meet you guys. So let me introduce myself. My name is Rizka Adriani. I am college at University State Islamic Syarif Hidayatullah, Jakarta, majoring at biology education, and now I am at 5th semester now. So why I'm in here? Because your teacher cannot teaching you today, so I am gonna tell you about the biology topic about the virus. So before we start, let me take your attendance list. Number 1, Alifa. Okay. Farah, Nabila, Dinda. Check all students' attendance until the last student number. Okay, thank you. So, no one absent today, right? So, it is our first meeting at the biology class. I wanna asking you for watching my videos about the virus. So, please pay attention to that video because after you watching the video, I wanna give you some question. Okay, let's start. So dear all my students, this video will be very interesting. So remember, when going viral was a good thing, what the meaning of that? That is a virus. Okay, viruses can spread rapidly, but they don't do it by themselves. So how? They hijack cells of living organisms and force the cells to generate copies of the original virus. The copies then take over other host cells and the cycle continues. And then virus can be spread through the air from a sneeze or cough. Of course, you already have said that. Viruses can be spread by what? By carrier organism like mosquitoes. Okay. And then viruses can be spread through bodily fluids. Did you understand, my student? Next, viruses can be spread from the surface carrying viral particles. These particles in everywhere, right? They are tiny, about 2,000 times smaller than bacteria. Virus infect nearly every living thing, plants, animal, even bacteria. Okay, that's all my video. So next, in the next session. Thank you for you all because already have to watch the video. So I want to give you this question. What we can learn from that video? If you have an opinion, just raise your hand. I will choose you. Okay, you, please go ahead talk. Time for Nabila give her opinion about the video. Okay, thank you so much for Nabila because you already speaking about your opinion about that video, about that virus. Anyone wanna talk again? Raise your hand. Okay, so in your life, of course, you already and ever get a cold, get a fever. You may be sneezing or cough. This make your health is very awful. So what kind of thing that can cause the symptoms? Who wanna answer? Just raise your hand. Yeah, you. Virus? Yes, you're right. So what kind of virus is? Virus is very little things in this world. Virus have a general characteristic. The characteristic can be non-living things and living things. So the first is they can duplicate in the whole cell that a human or animal. But as non-living things, virus cannot be grow, develop, and then reproduction with itself. So the virus need a host. That's why virus have a characteristic non-living things and living things. Did anyone want to talk your knowledge maybe? If you want, if you tell this more over. Actually, in the viral cycle, virus have a two cycle. That is lytic and the second is lysogenic. Basically, virus has a six cycle. The first is attachment, the second penetration, and then encoding, release, and assembling. So about the cycle of virus, you can look my pic under my PowerPoint. This is. So the cycles give us information about steps in a model viral life cycle. The first is attachment. Second is penetration, encoding, targeting, gen expression, genome replication, variant assembly or maturation, and release of new infections of virus. For the first is attachment. So, in the most cases, specific attachment proteins on the surface of viruses bind to specific receptor on the surface of animal cells. Cellular receptors are usually either glycoproteins or glycolipids and have other function for the cell in addition to virus binding. Second is penetration. This penetration do this primarily by one of two mechanisms. The first mechanism is endocytosis, and the two is direct membrane fusion. Next step is encoding and targeting. With some virus, the genome is completely released from the capsid during or after penetration. That is known as encoding. In others, such as retrovirus and rerovirus, the first stage of the viral replication cycle, like a transcription or replication, you actually occur inside the capsid. The next is gen expression and genome replication. Viruses with DNA genomes almost have genomes that are similar to the host cell. That is, they are composed of double-stranded DNA and are therefore able to utilize host enzyme to express viral genes and replicate viral DNA. Viruses with RNA must replicate entirely in the cytoplasm of infected cells. Exceptions are orthomyxovirus and retrovirus. The last step is virus assembly and release. Once new viral genomes and protein have been produced, they are assembled into new variants. For example, for many viruses, the viral capsid is partially assembled. Capsid protein associate together into a capsid-like structure. The virus then boots through the membrane to give rise to enveloped viral particles. These particles can then go through additional maturation even to give rise to infection virus. In the case of virus that form on the plasma membrane, they can boot from the cell without causing cell disease. Other enveloped viruses, however, are lytic. This is important to remember this. Virus have a structure. This structure, I can show you the pic. This one. This is structure of virus. The first is head and then neck, collar, whisker, basal plate, tail fins, tail tube, and tail fiber. In the head, there is a capsid. The capsid is the protein cell that encloses the nucleic acid. With its enclosed nucleic acid, it is called the nucleocapsid. This cell is composed of protein organized in subunit known as capsomer. The capsid has three functions. The first, it protects the nucleic acid from digestion by enzyme. Second, contains special sites on its surface that allow the variant to attach to host cell. And the third, provides protein that enable the variant to penetrate the host cell membrane and, in some cases, to inject the infection's nucleic acid into the cell cytoplasm. Bacteriophage have a tail that function for attach to the host cell. Okay, after I give you the explanation about the virus, I have this assignment for you guys. So you should have to work this at a group. So I will give you. Student doing their group assignment. Okay student, the time is already finished to work this, so this is time for you and your group to presentation in the front of the class. Student presenting their discussion group's result. I would say thank you so much and appreciation to you all that have the presentation the group discussion with better. For today's class, this is a summarize. The first is virus is a small things or microscopic things. They can cause living things or non-living things. And the second, virus commonly have a structure. There is a head, body, and tail. Next is virus have a cycle in their life. There is a lytic cycle and a lysogenic cycle. Virus can make a very much disease in this world. So the human try to destroy this virus by a vaccine or anything else. So that's all today class. Thank you so much. I'm sorry for my mistake. Wassalamualaikum warahmatullahi wabarakatuh. Thank you for you guys already watching my video.